So I'm really delighted to be here today and share with us, with you, your recent results. You know, also delighted to be here with my long-standing friends, Daniel Louva, Alain Puissieux, Thomas Bartlett, and Stefano Piccolo. So in my presentation today, I will talk to you about the mechanism that regulate the different tumor transition states. And as you heard from Thomas, with this excellent introduction about EMT, that's going to be really the focus of my talk today. And as you know, within a given tumor, not all cells are equal. Some cells proliferate, whether other cells invade the tissue and give rise to metastasis. And so next slide. And so I'm going to talk today about one model, which is the skin squamous cell carcinoma. So it's the second most frequent skin cancer, and is due to mutation in the RAS and the p53 pathway. Next slide. And so some time ago, we tried to understand whether tumor heterogeneity could be linked to the cell of origin, as Daniel Louva in his presentation alluded to. And to do this, we activate the oncogene that give rise to the skin tumor, KRAS, G12D, and p53 deletion in two different cell lineage of the same tissue, the skin epidermis. So the interfollicular epidermis here in blue, and in green, the hair follicle stem cell and its progeny. Next slide. And what we found is that the interfollicular epidermal cells give rise to what we call a well-differentiated squamous cell carcinoma. So it's a tumor in which all tumor cells express epithelial markers such as K14 in red, and none of the cells express vimentin, the mesenchymal marker that Thomas talked to you before. And as you see, also you have this presence of keratin pearls, this squamous material. But in hair follicle-derived tumor, most of the tumor were consisting of what we call a mixed SCC in the middle of the slide, in which a part of the tumor express epithelial marker, K14, the previous slide. Yes. So there was a part of the tumor expressing epithelial marker and a part of the tumor expressing mesenchymal marker, vimentin. So that was, you know, a pathognomonic of EMT. Next slide. So to quantify this phenomena, we could go to the flow cytometry and gauge on the YFP tumor cells. And as you see, using EpCAM expression, which is a little bit also an epithelial marker, such as E-cadherin, you can see that in the middle of the slide, that the tumor cells can present a bimodal expression of EpCAM. A high expression of EpCAM, it's the epithelial part of the tumor, and on the left of the plot is the mesenchymal cells, the EMT tumor cells, that has lost EpCAM expression. And so when you quantify in the graph below about several hundred of those tumors, you can see that almost all tumor cells present some EpCAM negative tumor cells, undergo to some extent EMT, but the range of EMT vary enormously from one tumor to the others. And you have this big repartition of the proportion of EpCAM positive and EpCAM negative cells. Next slide. So what I show you here is, depending on the cell of origin of the tumors, the same oncogenic hits will give rise to two different tumor phenotypes, either the well-differentiated tumor phenotype when it arises from the interfollicular epidermis, and these EMT tumors when it arises from the hair follicle. Next slide. So this EMT has a lot of importance. And also, as Thomas showed you, and we you know, kind of confirm what he showed you with the ZEB1 knockout, is the EMT tumor cells. They present high proportion of lymph node and lung metastasis. Next slide. Whereas the other tumor type, which do not present EMT or present rarely EMT, present a very low frequency of lymph node and lung metastasis. And in the rare case that present lymph node and lung metastasis, we found that in the primary tumors, some cells hadn't undergo EMT, suggesting that in this mouse model, EMT is, or metastasis is invariably associated with EMT. I don't mean that all tumors and all cancer will use EMT to undergo metastasis, but at least in this model, it seems to be the case. Next slide. The reason to why EMT, has been very controversial for a long time in the field of metastasis is when the pathologists look at the lung or the lymph node metastasis, they found that they could not find any sign of EMT. But in this mouse model, as well, if you look at the differentiation of the tumors in the lung or lymph node metastasis, many of the metastases do not present a remnant of EMT marker expression, such as Diamantin. So consisting with the notion that the reversion from mesenchymal to epithelial is important for metastatic colonization, as Thomas has also told you in his talk. Next slide. So in addition to being important for metastasis, EMT is very important to mediate resistance to therapy. And in our model, when you treat these mice with cisplatin 5-FU, the standard chemotherapy given to squamous cell carcinoma, you see that the epithelial part of the tumor in blue here are very sensitive to chemotherapy, and they die within 24 hours after you give cisplatin 5-FU, whereas the red one, the EpCAM negative tumor cells, they are highly resistant. And they are not only highly resistant to cisplatin 5-FU, they are highly resistant to a broad range of chemotherapeutic reagent and agent that promote replicative stress. And so Maud in my lab had worked for several years to understand the mechanism by which these EMT tumor cells are resisting to therapy, and it's still, an ongoing study in my laboratory. And so I will present that to another occasion. Next slide. So one of the key questions that we had when we saw this EMT in this mouse model was, do EMT occur through a binary switch, or do EMT occur through distinct tumor transitional states? And when you just look at the EpCAM expression, it looks like a binary switch, but maybe hidden within these nice peak of EpCAM positive or EpCAM negative cells, maybe they are hidden a big part of tumor heterogeneity. Next slide. Next slide, yes. So to address this question, we screen a panel of cell surface marker and check whether the EpCAM positive tumor cells or the EpCAM negative tumor cells express an homogeneous or heterogeneous level of additional marker. Next slide. So we screened these 200 cell surface marker, and what we found, that the EpCAM positive tumor population were very homogeneous. Most of the tumor cells there express are stained homogeneously with these different cell surface markers that we screen. In sharp contrast, the EpCAM negative EMT tumor cells, they present very often a bimodal expression of this cell surface marker, suggesting they are not an homogeneous population, but they represent a diversity of population hidden in these EpCAM negative tumor cells. So next slide, we then 2 by 2, 3 by 3, screen all these cell surface markers, and we found that with 3 cell surface markers, which is CD51, CD61, and CD106, we could deconvolute most of the tumor heterogeneity we found in this tumor. And so no, these cell surface markers are not, completely random, actually. They were precedent in the field. So CD51 and CD61 are alpha-3 beta-3 integrin, and that has been previously shown to be expressed in cancer stem cells. And CD106 was VCAM1 and was previously reported by the group of Johan Massagé as a metastasis-initiating cell. And you'll see in a second that these markers are indeed very important for the metastatic and the EMT colonization. Yes, next slide. So having this cell surface marker, we can, isolate them and cytospin and check the expression of the different epithelial K14 or mesenchymal vimentin expression on those different populations. And to our great surprise, we found that at the beginning of the EMT process, which we call the triple negative CD106, none of the, or very few tumor cells had lost the expression of the epithelial marker, and they co-express K14 and vimentin within the same cell. And we call these cells hybrid EMT. Other people call that partial EMT or quasi-mesenchymal EMT. All these terms are referring to a tumor cells that co-express two transcriptional programs, the epithelial and the mesenchymal transcription program at the same time. Next slide. And I think one of the most important findings of this study was the fact that when we test this different population for their ability to give rise to lung metastasis about tail vein injection, we found that the most mesenchymal tumor cells, the triple positive tumor cells, were not at all the most metastatic cells in this population. And the most metastatic cells were actually the early hybrid EMT population. And that was, a really big surprise. But after this study, many different groups had found that indeed in different mouse model and with, much more powerful method to track the metastasis initiating, including lineage tracing, they confirmed that the hybrid EMT were indeed, the most metastatic population found within this tumor. Next slide. Another interesting finding was the fact that these different EMT population were not all mixed up within the tumor, and they were still localized in discrete region of the tumor. And you see here there's at least 4 different regions that can be seen by their morphology, by their co-expression of epithelial or mesenchymal marker, and by the amount of cell adhesion that you can find. Next slide. And when you stain the tumor stroma for within this different region, we were very intrigued by the fact that these different regions presenting different level of EMT present very different tumor microenvironment. And the very EMT population, they are very immune infiltrated with a lot of monocyte, macrophage, and they also present, more blood vessels and lymphatic vessels, potentially explaining the dissemination of this of the metastatic cells into the lymph nodes. Next slide. So what I show you here is that EMT is not, a binary process, as indeed composed of different tumor states that present different tumor functions. The epithelial are the most proliferative. The early hybrid EMT state are the most metastatic, whereas the late EMT state are the most invasive population. And these different populations reside in different niche, and they are regulated by probably different type of transcription factors. But whether, in addition to this microenvironmental cue and transcriptional cue, would it be possible that different oncogenic mutation would promote different tumor states? That was, still an unresolved question. Next slide. So our attention was caught when we sequence skin cancer that has been arising from chemical-induced carcinogenesis. And we found that the mutation landscape found in this mouse skin SCC were very similar to the mutational landscape found in various human SCC population. And we were following up on trying to understand the function of these different oncogene or tumor suppressor gene that we found there, and for which the function was still, poorly characterized. And one of the genes that we were following up was FAT1. And FAT1 is a protocadherin. Next slide. And this protocadherin is heavily mutated in many different human cancer, and is the second most frequently mutated gene in head and neck squamous cell carcinoma, is among the most frequently mutated gene in lung squamous cell carcinoma, It's extremely frequently mutated in oral carcinoma, and in pan cancer, it really ranked among the top mutated genes. So it was, a tumor driver for which the function was very poorly characterized. Next slide. So we collaborate with Françoise Elschenberger who generate the FAT1 flux allele, and we obtained these mice from her, and we did the conditional deletion of FAT1 in the skin epidermis. And so as it was anticipated, when you mutated FAT1 in the skin, first there was no alteration of homeostasis, but when we treat these mice with chemical-induced skin cancer, the proportion of the tumor and their timing of appearance were dramatically accelerated and increased upon FAT1 deletion, consistent with the tumor suppressor gene function of FAT1. Next slide. But more interestingly and more importantly, when we inactivated FAT1 in benign skin tumor, these tumors were rapidly transformed into a squamous, invasive squamous cell carcinoma. Next slide. When we check the histological characterization of this transformation, we realized that within 2 weeks following FAT1 deletion, there was an effraction of the basal lamina, a disorganization of the tumor shape, and a block of differentiation, which is characteristic of the benign tumor. Next slide. But much more surprisingly and much more interestingly, we found that these very invasive tumors, they all present signs of epithelial to mesenchymal transition, but not again a full epithelial to mesenchymal transition. This hybrid EMT transition in which the cells co-express K14 and vimentin, the epithelial and the mesenchymal program at the same time. Whereas the control mice in this mouse model, they rarely develop invasive hybrid EMT tumors or even late EMT tumors. Next slide. So similarly to what I show you with the genetic tumor, when we check EpCAM expression, there was a bimodal expression of these of the tumor cells, of EpCAM expression in these tumor cells, with a lot of them block into this early EMT population, the triple negative of the CD106, and when we cytospin them, the EpCAM negative population were all hybrid EMT cells with the co-expression of K14 and vimentin. Next slide. And in those tumors, this hybrid EMT population were associated with spontaneous lymph node and lung metastasis at much higher frequency than in the control mice. Next slide. So I'm not going to go into the detail because it has been recently published, this study, but we try to understand how a transmembrane protein can lead to the activation of these hybrid EMT states, and ZEB1 is among the key factors that promote the mesenchymal expression, and we found that SOX2 was very important to promote the epithelial transcriptional program. And we really kind of nailed down the whole signaling cascade that leads to the activation of this stable EMT state. And we found that the caveolinin kinase 2 gets activated, phosphorylated Src and Yes kinase, which in turn were important for the translocation of yapentase to the nucleus and the activation of ZEB1 expression. And very interestingly, following the publication of this study, different groups has also found that caveolinin kinase 2 and calcium signaling was important for promoting hybrid EMT states in pancreatic cancer. That's the group of Ben Stangers, and the group of Barry Thompson in Australia has found also that the Src and Yes kinase were very important for the translocation and yapentase to the nucleus, suggesting that the mechanism that I'm suggesting that occurs here can be a generalized mechanism by which hybrid EMT state can be induced. Also, using this, by cracking this signaling pathway, we uncover tumor vulnerability in those hybrid FAT1 mutated tumor cells, and we found that they were exquisitely sensitive to SRC inhibitors. And so now we are trying to see whether, less toxic SRC inhibitor can be used to treat PDX cancer patient growing into immunodeficient mice. And if the case, the FAT1 tumor are indeed more sensitive, we were thinking that we could maybe think about moving that to a human patient. Next slide. So now I'm going to show you how, what we have learned from this study can be used to target EMT tumor plasticity and potentially having an impact on the tumor treatment. And this is it's been a wonderful collaboration with the group of Patrick Mellen and Agnès Bernet. So Patrick is a long-standing friend of mine, and so we were very happy to be able to collaborate. So Patrick has, uncovered the role of natrin in tumor regenesis and start to generate a blocking antibody against natrin-1 that is used now in clinical phase 2A, 2B to treat cancer patients. And so this study has been done by a PhD student that was in Patrick's lab and joined my lab a few years ago, Justine Langrand, and with Georgina Pastuchenko, a former postdoc in my lab, and Sebastian Van Neutem, a very talented bioinformatician. So next slide. So the story begins when we found that in a different cell population that had undergone EMT, natrin-1 was highly upregulated in this different population. And so, I contact Patrick and say, well, it looks very interesting here. Natrin-1 is highly upregulated in the tumor cells. Should we try to see inhibition of natrin-1 can impair EMT in this process? Next slide. So we treated the mice that developed this spontaneous EMT in the KRAS and p53 deleted model with an anti-natrin-1 antibody that is used in clinical trial. And we found that a little bit like Thomas has told you that, we could also decrease tumor initiation, but in the tumor that had been initiated, the proportion of positive tumor cells, so the epithelial tumor cell, was much higher in the anti-natrin treated group as compared to the control group, suggesting that anti-natrin administration prevent or at least inhibit EMT in this model. Next slide. This inhibition of EMT was associated with the reduction of spontaneous lung metastasis. Next slide. But also, and more importantly for the therapy, is the anti-natrin-1 treatment sensitize the tumor cells to cisplatin 5-FU, so to the standard chemotherapeutical reagent that is used to treat squamous cell carcinoma. Next slide. So when we did single cell sequencing to assess how these different tumor states were, impacted by the anti-natrin treatment, this is the control group. So, as I told you, with our monoclonal antibody, we found using clustering of single cell analysis here, the different tumor state that I present to you. In green, you have the early, the epithelial state, the one that express EpCAM, keratin 14, and do not express vimentin. You have this early hybrid EMT state with a loss EpCAM expression, continue to express K14, but co-express vimentin. And then you have this kind of late hybrid EMT in which you don't have that many K14 expression here, but you have another epithelial marker, K8 expression that is quite high, and the co-expression of vimentin. And then you have the late EMT population in which you lost keratin 8 expression, you only express vimentin and other markers such as PDGFR. Next slide. When you do the same analysis with the anti-natrin-1 treated animals, you see that the proportion of these different states were completely changed. And you had much more epithelial state, in this model, a tiny bit of early hybrid state, still some, late hybrid EMT population, but very, very, very few of this late late EMT population that are fully EMT. So clearly, pharmacological inhibition of EMT present the late EMT even. So when we do lineage trajectory, next slide, with different programs, including slingshot analysis here, it reveals that there's at least 2 lineage trajectory during EMT in these models, and the epithelial cells can go either toward this late hybrid EMT population or the late EMT population, and with all the different markers that I show you. But in contrast, next slide, when you treat it with the anti-neutrin monoclonal antibodies, you have also 2 different trajectory, but not anymore toward this late EMT population, but you have the appearance of another epithelial population which express high level of specific markers such as vitamin D receptor or OCT transcription factor that are highly epithelial, and you are moving toward this hybrid EMT population, but much less or not, almost not at all to this late EMT state. Next slide. So now with all these different markers that we found using the single cell analysis there, we could localize these different tumor states in situ and really confirm what we found following the anti-neutrin treatment, that this disappearance of the tumor state that express PDGFR and this EMT transition is stuck into this hybrid state that co-express the keratin 8 expression. Next slide. Yes, so in the last part of this talk, I will briefly mention, in less than 5 minutes, another story that related to the tumor state that mediate malignant transition. And here I need to disclose that I'm co-founder of a spin-off company that's called ChromaCure, in which we develop first-in-class molecule targeting the receptor that are mediating this malignant transition. Next slide. So the question arises 10 years ago in my lab we were wondering, what are the non-genetic factors that promote the transition from benign papilloma to malignant squamous cell carcinoma and skin cancer. And Federico Mori, next slide, found that among the genes that were the most upregulated in this malignant state was a nuclear orphan receptor called NR2F2, which was expressed exclusively on malignant SCC. And in benign papilloma, NR2F2 is only expressed in the endothelial cells of the tumor. Next slide. So we did, as we very often do in the lab, genetic loss of function of NR2F2, and loss of NR2F2 absolutely do not prevent tumor initiation, but it blocks all the tumors in their benign stage, and we could never see malignant tumor arising from fully deleted NR2F2 tumor. Next slide. When we did the reverse experiment in which we were overexpressing NR2F2, we found that this benign tumor, they change their morphology, begin to express a marker of invasion and early EMT states such as keratin 8, next slide, and the fraction of the basal lamina and acquiring malignant feature. Next slide. So when we check the tumor propagating cell frequency, which related to tumor stemness, when we delete NR2F2, there was a major reduction of the tumor propagation capacity of the tumor cells, both in the mouse and the human cancer, following NR2F2 deletion, suggesting that NR2F2 promote tumor stemness in these models. Next slide. But much more importantly, next slide. Yes. Much more importantly, for the therapeutical application, when we delete NR2F2 in already fully formed invasive tumor, this tumor regress very rapidly, and within 3 to 4 weeks, they are completely gone. Next slide. And something that was very interesting is when we delete NR2F2 and the tumor shrinks, now all of a sudden, they begin to re-express marker of benign tumor and marker of terminal differentiation, such as keratin 10 expression. Next slide. And when you look at H&E, you transform this tumor into a completely differentiated tumor mass, full of keratin materials. And this keratinization that you see here in pink with this KP there. And so, next slide. This differentiation reminds us a little bit the situation that is found when you treat it as Hugues de T and colleague has found some time ago, that when you treat it some type of leukemia with derivative of retinoic acid, which also target nuclear receptor, you promote the differentiation of the blast into a terminally differentiated material. And that's what, we really got interested into this gene, and because we knew that it's potentially druggable and we can promote tumor differentiation. So we also checked the mechanism that are regulating this differentiation, and so following gain and loss of function, we found that there was a modulation of cancer stemness, of collagen, extracellular matrix, and EMT and tumor progression that were down-regulate following the deletion or upregulate following the gain of function. Next slide. And for example, if you check the marker and the gene that Thomas showed you the key importance in tumor like ZEB1, they were massively upregulated following gain of function and were down-regulated following tumor loss of function. Next slide. So altogether, what we found is NR2F2 is very important for regulating this tumor malignant state, and it do so by regulating the function of different programs. It sustains the proliferative potential and the cancer stemness. It represses differentiation. It represses apoptosis. It represses the invasion of immune cells. And so when you remove NR2F2, there's a massive recruitment of neutrophils and other immune cell type in the tumor. And also it promotes tumor progression and EMT. Next slide. With that, I thank all the people that had done all this very interesting work in my lab. And so the first part of the study was Mathilde Latilde and Dalina Sarr. The second part on EMT and metastasis was a PhD student, Tanya Revenko. The hybrid EMT states and metastasis and FAT1 was the work of Eugenia Pastuchenko, which is now has her own lab and is assistant professor here in Belgium. And the last part is been done by Federico Mori, which is now group leader at Boehringer Ingelheim back in Vienna. Thank you very much for your attention. I also, sincerely apologize for all the trouble that I caused with my technical problem initially during for the presentation, but I'm very happy that, I could finally give you this talk, and we'll be very happy now to answer your question.
Okay, thank you very much for this great talk. I'm going to start with some questions about the two kind of therapeutical strategies that you propose with NETRIN, targeting NETRIN and NRF2. What would be for you the best combination or the settings because of this plasticity? Does it mean that you're going to need to treat always the patients? What is your point of view?
So, I think the recruitment of the patient, that's what Netris Pharma is doing. And, me, I'm really the basic researcher here. And I was the group who, kind of pushed this new concept of netrin and EMT. So following our discovery here, I know that the Netris Pharma has changed indeed the recruitment strategy and now recruited more patients that present tumors with some sign of EMT, and including such as I think uterus carcinoma and potentially other tumor with EMT feature. Indeed, so and now I think they also had an ongoing studies combining netrin antibody administration and chemotherapy in those patients they recruit.
Yeah, and for NRF2, so what would be the ideal combination? I mean, to target the.
So that NR2F2, and that's why also the very interesting thing that for this receptor. First, there's a very broad range of tumor actually that express NR2F2. There's not only squamous cell carcinoma. So NR2F2 is expressed in about 30% of lung adenocarcinoma. It's expressed in about 30% of pancreatic cancer. It's expressed in about 30% of melanoma, colorectal cancer, really to a broad range of solid tumor for which there's a medical need. So, I'm not completely sure that NR2F2 inhibitor would cure a patient in a monotherapeutical setting. In cancer, it's very often combination therapy. It's not because it won't cure a patient with monotherapy that we cannot potentially observe some clinical effect, a partial regression or other encouraging signs like this. But probably we will need to use a combination with either chemotherapy or with potentially immunotherapy, because I told you inhibition of NR2F2 lead to a massive tumor infiltration with immune cells. So we can potentially improve immunotherapy, and we are trying to test all these different possibilities in preclinical model and with the different compounds that ChromaCure had developed over the years.
But is it irreversible? It means that when you stop the tamoxifen or whatever, or the treatment...
in the mouse model that I present you, indeed is irreversible. So we have follow, tamoxifen is given acutely for a week, and then we have, follow the mice until, several months, and the tumor never regrow. And so some little tumor mass that was there where, when we cut them, there was no viable cells, it was just terminal differentiated material that we found there.
Okay. Concerning the story about FAT1 that I found really interesting, is it possible to rescue the phenotype when you have a tumor with this kind of mutation? Can you imagine to rescue the phenotype?
So what we try to do is to see whether restoring, so for example, overexpressing icadherin kind of restore the transcriptional program. The answer was no. Icadherin alone, overexpression, doesn't really restore the thing. Can we target preferentially this state? The answer is yes. With SRC inhibition, we have at least 1 log to 1 log and a half differences in sensitivity of what FAT1 mutated cells with SRC inhibitor. Kinase 2 inhibitor, is much less potent and much less specific. But also CAM2 inhibitor were showing this preferential killing of FAT1 mutated cells. So yes, we think that there's some lights there, and patients that present FAT1 mutation may benefit from SRC inhibition. And there's, the initial SRC inhibitor were a bit toxic, and now the people in Edinburgh at the Cancer Research UK had developed a very potent and very specific and non-toxic SRC inhibitor that we receive, and we are trying to see whether this can, change the response in a human patient direct xenotransplantation.
So, and in terms of,
there is time for 1 or 2 questions.
Okay, so maybe I can ask the last question. In terms of this inhibitor, ideally it would be given before the metastasis stages then. it would be to prevent, because
So indeed, prevent, we may hope to prevent, but also when we graft tumors and we treat them with the SRC inhibitor, the tumor, the cells present, the mice present tumor stasis at least, so, whereas in the control group, the tumor was still growing with this SRC inhibitor. So, you can impact on the tumor grow, and we didn't check specifically the metastasis, but, I imagine that potentially the metastasis may respond as the primary tumor, but that indeed, you're right, needs to be determined directly.
Okay, thank you very much, Dr. Blancpain and Professor Brablet, for this session. That was really interesting. And then